This is a really brief little lecture describing how we can use monomers, such as the ones we talked about before, like monosaccharides, fatty acids, amino acids, and nucleotides, to begin to build bigger molecules, these things we call dimers and polymers, such as starch and triglycerides and proteins and DNA and RNA, some of those big guys. Now, as you go through this lecture, I want you to think about what might be happening energetically in each of these reaction types. Is each reaction spontaneous or non-spontaneous? Is it exergonic or endergonic? Are we capturing energy or are we releasing energy that can do work other places? The building blocks of organelles and cells are these large organic molecules that we call macromolecules. Now, how are those macromolecules put together? Well, in general, all of the large macromolecules are going to be made out of small molecules called monomers. So when you think about a monomer, maybe think about one Lego block. All Lego creations are made out of Lego blocks, and one single little block, one small molecule, is called a monomer. If we put two of these blocks together, we might have something called a dimer. So these two blocks by themselves, we might call that a dimer, di for two. Two small molecules put together, we call a dimer. If we put more than two of these together, we get a larger molecule called a polymer. So a polymer would be all three of these put together. Polymers can consist of hundreds or even thousands of the monomer subunits put together. Now, different types of biomolecules are going to have different monomers, dimers, and polymers, and we'll go through that as we work through each group. We put monomer subunits together into dimers and polymers using a special reaction known as the dehydration synthesis reaction, also known as condensation. Now, it does have a big, scary sounding name, but really this name tells you what's going on in the reaction. So let's take a look at it. Here we have two monomer subunits. These are two small carbon rings. And notice that each of these has an OH, a hydroxyl group sticking out. We can use these hydroxyl groups to put these monomers together into one single dimer. And so that's what this reaction is all about. Now think about the words. What does it mean to dehydrate something? Well, of course, that means to remove water. And so that's what we're going to do. On one side, this monomer is going to give up a hydrogen. On the other side, this monomer is going to give up a hydroxyl group. And that's going to open up the bonds to put these two guys together, which you see has happened here. So you get rid of an H, you take off an OH, those two guys get together and make water. This is why it's called a dehydration reaction, because water is a byproduct. It comes out. And by doing so, you can synthesize a dimer or a polymer. The opposite of building a polymer is, of course, breaking one down. And that's what happens in this reaction known as hydrolysis or hydrolysis. This is the reaction that happens in your stomach and in your intestines when you break down your food, for instance. Now, in this reaction, we're going to have a polymer or a dimer, and it's going to get broken down into monomer subunits. So how does this work? Well, hydrolysis is really dehydration synthesis in reverse. So what's going to happen is that a water molecule is going to come in here. A hydrogen from one side of the water is going to go one way. The hydroxyl group is going to go the other way, and it's going to push this bond around this oxygen apart. So here you can see that the hydrogen has joined on this side, and the hydroxyl group has joined over here on this side, and it's separated these two molecules into their monomers. The word hydrolysis means water breaking. So hydrolysis or hydrolysis.